Okay, everyone. This next recording is on lipid metabolism. Okay, so in lipid metabolism we have catabolic processes and anabolic processes. Lipid catabolism is also known as lipolysis. Okay, as we know, triglycerides are the main type of dietary fat and the most common type of lipid in the body. Like cholesterol, triglycerides can be made in the liver and obtained from the diet. Okay, so we know that fat is mainly stored as triglycerides in our adipose tissues. Okay, when glucose levels are low, which occurs when we're fasting, right? We have to use our reserves. Okay, so our reserves are glycogen, which is stored in liver and skeletal muscle. Okay, and glycogen can be broken down into glucose to maintain those levels. Okay, but we can also use fatty acids. Okay, so what happens is that when we are under stress and we're fasting, the pancreas releases glucagon, and our adrenal medulla releases epinephrine. Okay, these hormones, they signal, they bind to receptors on adipocytes and basically signal adipocytes to break down their triglycerides. And they do that. So these hormones basically activate the lipases, lipases that we have inside adipocytes. And basically, these lipases, which are, in general, there's 3 of them there, it's going to cause triglycerides to be broken down into glycerol and fatty acids. So glycerol and fatty acids, they're going to get into our bloodstream. Okay. Now, glycerol is more, water soluble than fatty acids. So fatty acids need to be transported. The main transport protein for fatty acids is albumin. So basically, when fatty acids get to their target, which can be mainly muscle, like cardiac muscle, skeletal muscle, or liver, then these fatty acids can be utilized. Okay, basically, these fatty acids will be transported into the mitochondria, and then when they're in the mitochondria, then you can break them down by 2 carbon molecules, basically. So let me show you how that's done. But breakdown of fatty acids in the mitochondria, we call that beta oxidation. We call that beta oxidation, as you see that word over here. Okay. Now, let me show you something else. Okay. Here is the end of a fatty acid. For example, this is the carboxylic acid group here. Notice that we have a carbon here attached to a double bond O and an alcohol, an OH. So basically, this is a carboxyl group or carboxylic acid group. We have this carbon here. The next carbon is called the alpha carbon, and the carbon after that is the beta carbon. So basically, during beta oxidation, we are splitting. We're making a cut right here. Okay, we're making a cut right here, and we're making a molecule that has 2 carbons. Okay, and we call that acetyl-CoA. All right. So let me show you in the notes here and in textbooks. Usually they have a simplified version of how this takes place. Okay. So let me show you what's happening here. So basically, in the cytoplasm of cells that are needed to, this extra energy, in the cytoplasm, what you're going to have is, the fatty acid. Okay, so you have the fatty acid. Now, the fatty acid, this one in particular, has 18 carbons. If we count the carbons here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So this is an 18 carbon fatty acid, okay? So this is in the cytoplasm of the cells, like this. It could be cardiac muscle, it could be the liver, it could be skeletal muscle, okay? So basically, and you learn about that, you learn about that in muscle tissue in AMP1, like what happened when a skeletal muscle was resting, when the skeletal muscle was doing, was engaged in motor activity, and when it was doing peak muscle contraction. So you learn about that in AMP1. So what happens here in the first step, in order for us to for this fatty acid to be taken into the mitochondria, we need to invest energy here. So in the beginning, we're investing ATP. Okay, once we're investing ATP because we need to add a bond. We need to form a bond here. So we're adding, a coenzyme A to the end here. So, and that requires energy. So once we do that, then it gets transported into the mitochondria, in the matrix of the mitochondria, okay? There's going to be a series of reactions that are going to take place until you get to the very last step. In the very last step, okay, is where you're going to have an enzyme that is going to split the molecule here and cut the molecule by 2 carbons, okay? And you can see here that it has already been done, like, for example, over here. And then we have, we have acetyl-CoA right here, okay? Now, acetyl-CoA can be taken into the Krebs cycle, okay? The Krebs cycle, or enters the Krebs cycle, and from each acetyl-CoA, we can generate 12 ATPs, okay? Now, in this process, from the time that the fatty acid was taken into the mitochondria and to the time where it was split into 2 carbons, there were high energy intermediates that were generated here. We have NADH, we have FADH2, right? But notice that this is an oxidative process. Notice that NAD and FAD are oxidizing agents. So basically they're oxidizing the substrate and becoming reduced. So once they become reduced, they're energized. They're taking electrons from the substrates here. So these NADH and FADH2s, they're going to be heading into the electron transport system. And from there, we're going to be making ATP because this system is coupled with oxidative phosphorylation, as we learned when we talked about carbohydrate metabolism. Okay. Now, the important thing here is that how many cuts are made here from an 18 carbon fatty acid? We're making 1 cut here to generate a molecule of a 2 carbon molecule, right? So we're making 1, 2, 3, 4, 5, 6, 7, 8. So we're making 8 cuts. And so that's equivalent to 8 rounds of beta oxidation. Okay, 8 rounds. All right. So that means that we're going to be generating 8 molecules of NADH and 8 molecules of FADH2s. Okay. And also we are generating 9 molecules of acetyl-CoA. Why 9? Because we have 2. You have like 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 molecules of acetyl-CoA. Now, when you multiply 9 times 12, you're going to get a certain number, okay? And that number is 108 ATPs, okay? Now, we learned from prior video that one NADH gives you 3 ATPs, okay? But we generated 8 of them, so 8 times 3 is 24, right? And then FADH2, one of them yields 2 ATPs. So we're making 8, so 8 times 2 is 16. When we add everything here, we generate 148 ATP molecules, okay? Now, we do have to subtract you know, the ATPs that were invested in the beginning. So at the end, we're making 146 ATPs from an 18 carbon fatty acid. Okay, do you see the difference? You see that from one glucose, you get, you know, 36 ATPs, but from 18 carbon and 18 carbon fatty acid, we can make 146 ATP molecules. So you can see that fats, you know, from fat, we can make more energy. It's just that fats are not as accessible as carbohydrates are, okay? So, and that is there, and it takes time, you know, for us to get energy from fat. So at the end here, so what is the takeaway? The takeaway is that when cells need energy and you don't have, you're running on resources that are carbohydrates. When you're running out of carbohydrates, and you're fasting, and you know maybe the person has diabetes type 1, where they don't make insulin, and glucose is not going to be able to get into the cell. So therefore, you need to get energy somehow, you know, from something else, and it will be from fatty acids. So basically, you'll send a signal to the fat cells so that they can liberate, you know, the glycerol and the fatty acids. And then these fatty acids are transported by albumins to the cells that need it, that need this energy. And then through beta oxidation, we're going to generate acetyl-CoA, and from acetyl-CoA, we're going to get lots of ATP. You know, glycerol, which is another product of of the breakdown of triglycerides. Glycerol can also be used, can also be turned into glucose by the liver in a process known as gluconeogenesis. Okay, so let's continue here. Okay, we're almost done here because that's the main point is what I just said. Okay, you know, beta oxidation is the breakdown of fatty acids, and this takes place in the mitochondria primarily, and we're making ATP from it. Okay. Now we can also, you know, in times when we don't need the extra energy, we can build on fats. Okay, and that's called lipogenesis. Okay, so you know, in here I talk about lipogenesis. You know, to make fatty acids, you acetyl-CoA is used. You know, the acetyl-CoA is generated through carbohydrate metabolism. Also, when we are taking fatty acids into, you know, basically, you know, we can also make lipids. We can make. Let's see here. We can. So because lipids, amino acids, and carbohydrates can be converted to acetyl-CoA, acetyl-CoA can be used to make fatty acids. Yes. So yeah, so acetyl-CoA, again, you know, I lost train of thought for a moment, but acetyl-CoA can be used to make fatty acids, you know, in general. DHAP can be used to make glycerol. You know, I don't want to get into this topic here because basically, I do want you to know that fatty acids can be made from acetyl-CoA, and acetyl-CoA can be generated from fatty acids as well. Okay, that's the takeaway. Let's continue here. You know, most lipids, they're not soluble in water, so they're transported by lipoproteins. Fatty acids, however, they're generally transported by albumin. You know, there is other so lipids that are not soluble in water, like cholesterol, for example, can be transported, you know, by lipoproteins. And there is in here, there is a number of them. There is 5 classes of lipoproteins. These are proteins that transport lipids. Okay, the largest of them are chylomicrons. So chylomicrons can transport lipids from the small intestine to the bloodstream. Okay. We can also transport triglycerides from the liver to the bloodstream by very low density lipoproteins. But I want you to know kilomicrons. Kilomicrons is how we can, it helps us to transport lipids from our, small intestine to the bloodstream. And I want you to know the LDLs and HDLs. Okay, so what they do. So let me show you here what they do. By the way, here in this image, you can see the work of kilomicrons. You can see that the intestinal mucosa in the intestinal tract here, we have kilomicrons. We have a fat here, and then it's packaged into kilomicrons. Kilomicrons are taken by lacteals, which are capillaries, lymphatic capillaries in the small intestine. They eventually, these kilomicrons, they into the thoracic duct and into the bloodstream. Okay, so you can see that kilomicrons are transporting lipids from the intestinal tract to the bloodstream. Okay. All right. Let's see. And then once in the bloodstream, they can get to target organs. Okay, they can get to target organs. Let me show you something here. Now they talk about lipoprotein lipase, but I don't want to get into that for the time that we have remaining. I want you to know what LDL is doing. LDL, these are low density lipoproteins. They're transporting cholesterol from the liver to your peripheral tissues. So they refer to LDL as bad cholesterol. Like LDL cholesterol is bad cholesterol. You don't want this to be high. Traditionally that's what they have been. That's what it has been. What we have known, okay, and that still remains, okay. Now, HDL is high density lipoprotein. This one transports cholesterol from the tissues, from the peripheral cells, from the tissues, back to the liver for elimination in the bile. So HDL is the one that they tell you to keep high. You want to keep HDL high and LDL, low. So basically, there are numbers here, associated with LDL and HDL, specific numbers. You're going to learn that more like in pathophysiology. Okay, there's specific numbers here that need to be met. Okay, let's now I want you, what they're taking now as a risk factor for stroke is the ratio between triglycerides and HDL. So triglycerides, they need to be, less than 150. So triglycerides, when they're elevated and your HDL is low, that is a risk factor. The ratio is going to be high for having a stroke. You're going to be more at risk. Okay, so you want to bring down your triglycerides and pick up your HDL. HDL can be increased by exercise. If you exercise, and also, if you drink like a cup of wine after meals, that can also raise your HDL. Okay, but if you are sedentary and you're not working out, you're not exercising, that's your LDL is going to rise and your HDL is going to drop. Okay. So, so basically, that's what I wanted to say. This ratio, triglyceride HDL ratio, should be optimal, should be less than 2, but it can range between 4. If it goes, if it's 4 or lower, right? That's good, but optimal is 2. But if it's 6 or higher, that's a high risk factor for having a stroke. Okay. And that's what I wanted you to know, just to have a little bit of an idea what is lipid catabolism. Okay. So to make fatty acids, you need acetyl-CoA. To make energy, you need to take fatty acids into the mitochondria to generate acetyl-CoA to make ATP. Okay. So there is lipid catabolism and there is lipid anabolism. Lipid anabolism is when you're building lipids. Catabolism is when you're breaking lipids. Okay, so we're emphasizing more on breaking down lipids in times when you're fasting, when your glucose levels are low, when you're stressed out, when epinephrine and glucagon will be released. When your glucose levels are low, glucagon is released by the alpha cells of the pancreas, and your sympathetic nervous system, you're going to be stressed out. It's going to be activated, and that's going to trigger the adrenal medulla to release epinephrine in higher amounts, and they're going to activate lipases in fat cells, causing them to release their fatty acids and glycerol, okay, into the bloodstream. And again, glycerol is more water soluble than fatty acids. Fatty acids need to be transported by albumins. Albumins get to the organs that are going to be needing these fatty acids, and these fatty acids are going to be taken into the mitochondria, but energy needs to be invested for that. And then once they're in the mitochondria, you're generating NADHs, FADH2s, and ATP. Okay, so that's how that works. Okay, so I'll leave it there, and I'll make another video on protein metabolism, and we will be done with this topic. Okay, so see you in the next video.